A few years ago, a team of researchers, including scientists at Stanford University, working alongside colleagues studying cellular metabolism, turned their attention to a drink that has been part of Japanese tradition for centuries: matcha green tea. And what they found sparked genuine excitement across the cancer research community. Unlike regular brewed green tea, where the leaves are steeped and then discarded, matcha is made by grinding the entire tea leaf into a fine powder that is then whisked directly into water. Meaning, when you drink matcha, you are consuming the whole leaf itself, rather than simply an extraction from it. This distinction turns out to matter significantly for the concentration of certain beneficial compounds, and it's part of why matcha specifically, rather than green tea more broadly, has become a particular focus of recent laboratory research, examining effects on some of the most stubborn and dangerous cells involved in cancer, a specific subpopulation known as cancer stem cells. Today, we're going to walk through what this research actually found, why cancer stem cells represent such a significant target in modern cancer research, what compounds in matcha researchers believe are responsible for the effects observed, and importantly, how to think about this kind of early-stage laboratory research in proper scientific context. To understand why this research matters, it helps to understand what cancer stem cells actually are, and why researchers have become so focused on them in recent years, since this concept represents a genuinely important shift in how cancer biology is understood. For a long time, cancer was thought of as a relatively uniform mass of abnormal cells, all roughly equivalent in their behavior and vulnerability to treatment. Research over the past 2 decades has revealed a more complex picture. Within many tumors exists a smaller subpopulation of cells, called cancer stem cells, that share certain properties with normal stem cells found elsewhere in the body, particularly the ability to self-renew, essentially replicating themselves indefinitely, and the ability to differentiate into the various other cell types that make up the bulk of a tumor. This matters enormously from a treatment standpoint, because research has found that cancer stem cells tend to be disproportionately resistant to conventional chemotherapy and radiation, compared to the more differentiated cancer cells that make up the majority of a tumor's mass. Many standard cancer treatments work by targeting rapidly dividing cells, but cancer stem cells often exist in a relatively slow dividing or dormant state, allowing them to survive treatments that successfully eliminate the bulk of a tumor, only to later reactivate and drive cancer recurrence, sometimes months or years after a patient appeared to be in remission. This is part of why researchers increasingly believe that effective long-term cancer treatment may require specifically targeting this cancer stem cell population, not just the more numerous but less dangerous bulk tumor cells. And it's exactly this population that the matcha research specifically focused on. The Stanford-led research team, in a study published in the scientific journal Aging, examined how matcha green tea affected cultured breast cancer stem cells in laboratory conditions, using a research approach called metabolic phenotyping, which allows scientists to study how a treatment affects the internal metabolic state of cells, essentially examining how the cells are generating and using energy, rather than simply observing whether cells live or die. What the researchers found was that matcha exposure shifted the cancer stem cells toward what's called a quiescent metabolic state, essentially a slowed down, less metabolically active condition, and that this shift occurred at relatively low concentrations of matcha in the laboratory setting. Since cancer stem cells rely heavily on their metabolic flexibility and activity to support their self-renewal and eventual differentiation into cells that drive tumor growth and spread, pushing these cells into this quieter, less active state represents a genuinely significant finding from a mechanistic standpoint, since it suggests a pathway through which matcha compounds might interfere with the specific properties that make cancer stem cells so dangerous and treatment-resistant in the first place. Beyond this metabolic shift, the research team also found that matcha appeared to influence specific cell signaling pathways known to be involved in promoting and maintaining cancer stem cell populations, meaning the effects observed weren't limited to a general metabolic slowdown, but appeared connected to more specific molecular mechanisms relevant to how these particular cells maintain their dangerous stem-like properties. This finding connected matcha's effects to a broader body of laboratory research from the same research group and others, who have separately studied various natural compounds for their effects on these specific signaling pathways, including earlier research from some of the same Stanford-affiliated researchers, examining a compound found in Earl Grey tea called bergamot, which was found in related laboratory research to have effects on cancer cells and cholesterol metabolism, suggesting these researchers have been building a broader body of work examining how various natural plant compounds interact with cancer cell metabolism and signaling more generally, with matcha representing one of the more striking and widely discussed findings within that broader research program. So what specifically within matcha do researchers believe is responsible for these effects? And this is where the conversation connects to a compound that has actually been studied extensively across cancer research for several decades now, a catechin called epigallocatechin gallate, commonly abbreviated as EGCG, which represents the most abundant and most extensively researched of the polyphenol compounds found in green tea generally. What makes matcha particularly notable in this context is its concentration of this compound, since research measuring catechin content across different tea preparation methods has consistently found that matcha, because it involves consuming the entire ground leaf rather than a water-based extraction, contains meaningfully higher concentrations of EGCG and related catechins compared to standard brewed green tea made from steeped leaves. Some research suggesting concentrations several times higher depending on preparation method and comparison. EGCG itself has an extensive independent research history spanning laboratory studies, animal research, and some human clinical trials, examining its effects on cancer cells more broadly, not limited specifically to cancer stem cells. Laboratory research has found that EGCG appears to influence multiple processes relevant to cancer, including inducing a controlled form of cell death called apoptosis, specifically in cancer cells, while showing considerably less effect on normal, healthy cells at comparable concentrations. A selectivity that researchers find particularly encouraging since it suggests a genuine mechanism of action, rather than simply broad cellular toxicity that would be expected to harm healthy tissue as much as cancerous tissue. Research has also found that EGCG appears to have antioxidant properties relevant to reducing the kind of cellular damage that can contribute to cancer development in the first place, along with some evidence suggesting effects on the blood vessel formation, called angiogenesis, that growing tumors depend on to receive adequate blood supply, a process some research has found EGCG may help inhibit. Population-level research examining green tea consumption more broadly, separate from the specific laboratory studies on matcha and cancer stem cells, has added another layer of relevant context to this picture. Some of the more extensive population research in this area has come from Japan, where green tea consumption has historically been high enough to allow researchers to examine associations between consumption levels and cancer outcomes across large populations over extended time periods. This research has found associations between higher green tea consumption and reduced risk or delayed onset of certain cancer types, with some of the more striking findings coming from studies examining populations with particularly high consumption levels, generally described in this research as exceeding 10 cups of green tea daily, a level considerably higher than typical consumption patterns in many other countries, which is worth keeping in mind when considering how directly these population findings might translate to more moderate consumption levels more common elsewhere. It's genuinely important and intellectually honest to place this entire body of research in appropriate scientific context, because the matcha and cancer stem cell findings specifically represent laboratory research conducted on cultured cells, not human clinical trials. And this distinction matters enormously for how confidently any of these findings should be interpreted or applied. Laboratory research on cultured cells serves an essential and valuable purpose in the scientific process. It allows researchers to identify plausible mechanisms and generate hypotheses that can then be tested in more complex systems, first in animal models, and eventually, if the evidence continues to support it, in human clinical trials. But the jump from a compound affecting cancer stem cells in a laboratory dish to that same compound providing meaningful cancer prevention or treatment benefit in an actual living human body involves numerous additional factors that laboratory research alone cannot fully address, including how well the relevant compounds are actually absorbed when consumed as a beverage, how they're metabolized and distributed throughout the body, whether they reach cancer-relevant tissues in concentrations similar to what was used in laboratory testing, and how they interact with the vastly more complex biological environment of a whole human body compared to cells growing in a laboratory dish. This is not to dismiss the significance of the research. Laboratory findings like these represent genuinely important scientific progress and have historically formed the essential foundation for treatments that eventually proved effective in human trials, but rather to encourage appropriate calibration of expectations. Matcha green tea, based on current evidence, should be understood as a beverage with genuinely interesting and scientifically plausible properties relevant to cancer biology, supported by a reasonable body of both laboratory and population-level research, rather than as a proven cancer treatment or a guaranteed preventive measure, a distinction the researchers involved in this work have themselves been careful to acknowledge in how they've discussed their findings publicly. With that context established, it's worth discussing what a reasonable, evidence-informed approach to matcha consumption might look like for someone interested in incorporating it into their diet, based on genuine interest in this research, rather than either dismissing it entirely or treating it as a miracle intervention. Research on catechin absorption has found that these compounds are indeed bioavailable when consumed as a beverage, meaning they do enter the bloodstream and reach circulation, though absorption efficiency varies based on factors including whether matcha is consumed with food, since some research has found that certain proteins can bind to catechins and reduce their absorption, and individual differences in gut bacterial composition, which, as research in other areas of nutrition science has found, can influence how effectively various plant compounds are metabolized and utilized within the body. Preparation method also appears to matter based on available research, with matcha prepared using traditional whisking methods in water, rather than dissolved into large amounts of milk, which some research suggests may bind to and reduce absorption of catechins, similarly to the protein binding effect just mentioned, generally associated with higher measured catechin bioavailability in the research examining this specific question. Regular, moderate matcha consumption, incorporated as part of an overall varied diet, rather than relied upon as a singular protective measure, aligns reasonably well with the current state of this evidence, sitting alongside the broader dietary patterns that research across cancer prevention science more generally has found most consistently associated with reduced cancer risk. Patterns built around diverse plant foods, adequate fiber, limited processed food and alcohol, and maintained healthy weight and physical activity. Categories we've explored in other discussions, and which continue to represent the most robustly evidenced foundation of dietary cancer risk reduction, with matcha and its concentrated catechin content representing a genuinely interesting and scientifically supported addition to that broader foundation, rather than a replacement for it. Stepping back, what this research on matcha and cancer stem cells ultimately represents is science working exactly as it should. Careful laboratory investigation into a traditional beverage, conducted using sophisticated modern research techniques, uncovering plausible and genuinely interesting mechanisms through which specific compounds might interfere with some of the most treatment-resistant and dangerous cells involved in cancer development and recurrence. Whether this research eventually translates into clinical applications, perhaps concentrated catechin compounds used alongside conventional cancer treatments to specifically target the cancer stem cell populations that so often drive resistance and recurrence, remains to be established through the continued research process, including the animal studies and human clinical trials that would need to follow this kind of early-stage laboratory finding. What can be said with genuine confidence today is that matcha green tea represents a beverage backed by a meaningfully growing and scientifically credible body of research relevant to cancer biology, one worth understanding accurately, appreciating for what it currently demonstrates, and watching with genuine interest as this specific line of cancer stem cell research continues to develop in the years ahead. It's worth briefly touching on the broader landscape of natural compound research this matcha finding sits within, since it's far from an isolated curiosity in the cancer research world. Researchers studying various plant-derived compounds have identified quite a number of substances that show promising effects on cancer cells in laboratory settings, from curcumin found in turmeric, to resveratrol found in grapes and red wine, to sulforaphane found in cruciferous vegetables discussed in other contexts. And matcha's catechins take their place within this broader, active area of what researchers sometimes call phytochemical cancer research, What distinguishes the matcha and cancer stem cell findings specifically is the particular focus on this treatment-resistant cell population, rather than cancer cells more broadly, and the specific metabolic phenotyping approach the Stanford team used, which offered a more detailed mechanistic window into exactly how the observed effects were occurring, rather than simply reporting that cell numbers decreased following treatment. A methodological detail that gives the findings somewhat greater scientific weight within the laboratory research literature, even while the translation to clinical human benefit remains, as discussed, an open and ongoing question. It's also worth noting that matcha contains compounds beyond EGCG that have drawn separate research interest, including an amino acid called L-theanine, found in relatively high concentration in shade-grown tea leaves, like those used for matcha specifically, which has been studied primarily for its effects on relaxation and cognitive function rather than cancer biology directly, though some research has examined potential interactions between L-theanine and EGCG, with a few laboratory studies suggesting these compounds may work together in ways that affect their combined biological activity. An area of research that remains considerably less developed than the EGCG-focused work discussed throughout this conversation, but reflects the kind of compound interaction complexity that makes whole food and whole beverage research, as opposed to studying isolated compounds alone, a genuinely valuable complementary approach within this broader field. For anyone drawn to this research and considering matcha as part of their regular routine, the most scientifically grounded takeaway is a balanced one. This is a beverage with real, published, peer-reviewed research behind it, examining mechanisms genuinely relevant to some of the more dangerous and treatment-resistant aspects of cancer biology, and incorporating it as an enjoyable, regular part of an already health-supportive diet represents a reasonable choice grounded in legitimate science, while understanding clearly that no current evidence supports treating it as a replacement for established medical screening, treatment, or the broader lifestyle factors that remain the most thoroughly evidenced tools available for reducing cancer risk over the course of a lifetime.